All right, good morning. Second day, I'm going to give some update from the Tennessee's field stuff. So, um, Bode Okulu, most of you know, he moved to Australia. So I have not heard from him, but maybe he's sleeping now. So from his update, what he is going, maybe sometimes he will share with Oscar and will know there. But he already sampled all the rose sample he needed, so he might be working on the lab and what's going on that microbiome thing. So he will update sometimes. So yes, we talked about from the Oklahoma and other stuff. We have a lot of infected roses in the field. I'll share some good pictures here. If you really want to see a lot of infected roses, please visit Crossville, Tennessee. I'll show you all these beautiful infected roses. So field trials, we do the varietal screening nursery. So we have around 4,000 almost roses with along with the GWAS study. I'm going to share the data from 2024. And then we have already collected the rose sample for 2025. And I already rated the roses, all of them. So maybe the data will come for 25 later. And then the mite population study, my graduate student, Lina, will present about how the mite counts going on with her project. So from 2024, we collected almost around 35,100 roses, and then we do the qPCR, RT-qPCR, and I just saw the Dr. Ali's presentation. RNA3 is not detected there. All of our bases right from RNA3, right? So, but yeah. So we also record the RRD rosite and then the severity. So how many shoot per rosite and how many infected rosite shoot per plant. So we also record that. And then this year and last year, I also recorded the percentage of severity, the impact of the rosite in overall plant. So I have this data also. So here you can find 2024 RRD assessment. 35 plants were positive to RRD based upon the qPCR result as well as our visual assessment. So what happened in 2024, I have some undergrad or many, we have 4,000 roses to collect the samples. So we have many people there. So some of the confirmed positive 3x3 rated roses came negative to RT-PCR. I was really surprised what's going on there. I know that plant has symptoms. I rated that. And then I realized that we have many people to sample undergrad. And so maybe we, listening music and just pull the leaf. Maybe at that time, some of us may not realize the infected cane sample or non-infected. We already know many canes may have no symptoms, no virus there. So that's the reason. That's why I asked Caroline, hey, Caroline, go to the field again and sample that infected and non-infected roses. So all infected came to positive. Now, all these CT values are there. This is from 2024. So this dash there, no CT value means the plant was dead. We saw the symptoms, but that during the time of sampling. So you see these beautiful RRD infected plants there. So in 25, so I just, I have the data in 25, 66 plants showed the visible symptoms. Right? I recorded symptoms there. So we need to run the RT-qPCR on them. So we see more plant infected this year than compared to the last year. So this is all the genetic study and cultivar varietal screening nursery data of 24. So the GWAS study, so we have a 728 new cultivar for the GWAS study. Then in 2024, around, I think 11 plants were positive. Only one variety has symptoms. You can see in May 2024, the symptoms was like this, but all of them didn't have symptoms but came to positive from RT-qPCR. And I double check only these cultivar whether they have symptoms in 2025. Yes, they had symptoms in 2025. Some of them were dead because of the virus infection. You can see in the field also the symptoms in May 24, July 24, November 24. Look at that cultivar within a 5-6 months is completely dead. So even in the field, we can see these stuffs. So maybe that, we thought about maybe the translocation of carbohydrate during the hibernation, winter season or so. It shows within a high temperature, right? Or maybe that November has a little bit chilly, but it's plant died.
The question was, do you know what cultivar it is?
It's a good question. It's a good question.
It's a good positive control.
One slide. Is that
Living easy.
Living easy. Okay.
Living easy.
Got it. So it's a good positive control.
Thank you, Kevin.
Yeah.
Okay. This year, we got a really good symptoms in Jivas. You can see all of these canes has rosite, and some of the canes are really bushy. 3x3 rating. So you have seen this all already. So, and 62 plants showed visible symptoms. So 2024 we had 11. Only one showed symptoms and 11 positive. But this year we found 62 plants shows visible symptoms like this. At least one cane infected. All right, so that's what I have for the varietal screening and Jivas study. I want to give more time to my students. He has a really great slides and really good story to talk about. Okay, so now Elena, take it over.
Thank you, Dr. Ranabhat. Good morning, everyone. My name is Elena Pokrel, and I'm a graduate student working with Dr. Ranabhat at the University of Tennessee. I joined his lab last year as a PhD student. And being a newcomer to this wonderful group, I'm truly grateful to be here and to have the opportunity to present alongside all of you. So today I'm going to talk about my part of the project, which is field studies of Phyllococcus fructiferus mite population on rose-rosette disease susceptible and resistant cultivars. Since we have learned enough about rose-rosette disease yesterday in different sessions, I'll go straight into my part, which is the field study of the mites. We conducted this field study to compare mite populations and rose-rosette disease incidence across the resistant or susceptible rose cultivars, with the goal to determine if post-plant resistance to mites is a contributing element to rose-rosette disease resistance. So for that, we set up our field at UT Plateau Research and Education Center, which is in Crossville, Tennessee. This is how our field looks right now. So for our field, we did randomized complete block design with 5 blocks and 3 replications of each cultivar per block. We had 12 cultivars in total, which you can see towards the right side of this slide. We did this plantation on May of 2024. So after planting, we let the plants establish themselves in the field for a couple of months. And on August of 2024, we did the mite inoculation. So the mite inoculation procedure was pretty much similar to what Dr. Ali already mentioned it. We looked for infected rose plants, collected the infected branches, and clipped the infected branch into the healthy plants in our field. So after mite inoculation, our plan was to see if we could see the early establishment of the mites or early development of the symptoms in the field, because we have seen in the literatures that once the mites get very fevers after continuously feeding on the infected plants for around 5 days, they can transmit the virus as early as an hour. Also, if we look at the rose disease symptoms development, we have instances where symptoms were visible as early as a month or 6 months. So, and again, if we look at the mite dynamics, this mite has very short life cycle of around 7 to 12 days. So it has multiple generations within a season if it gets a favorable condition. So we wanted to see if by chance we see early establishment of the mites in the field, or if we are able to see the visible symptoms. So we looked into the fields on September and October after a couple of months of inoculating the mites. But at that point of time, there was no visible symptoms in the field, as well as when I collected samples for extracting mites, mites number were negligible, almost zero in all the samples I collected. So, okay. So, and after October, we ran into seasonality. As the temperature drops, mites activity as well as reproduction declines. Instead of actively feeding and dispersing viruses, mites undergo overwintering. On top of that, plants are also not pushing new tender growths, which are favorable. So putting all these things together, winter is not so favorable season for collecting the samples to collect mites. So we paused our sampling for throughout the winter and resumed in late spring when the warmer temperature as well as the flushes from new growth support mite activity as well as have the better chances of capturing the mites. I'm pressing something without knowing. Okay. So for collecting the samples, we focused on the preferred niches of Phyllococcus fructifilus, which included the flower buds and sepals, petiole stem interface and stipules, as well as the young leaves. So since it is fructifilus, we mostly focused on the buds. Those cultivars that didn't have buds, we collected enough of the tissue samples from other tissues. So after collecting the sample, the next step was to extract mite. I'll briefly talk about this protocol, not in detail. So for extracting mites, I took 5 grams of the tissues, added a little bit of soap and 250 ml of 7% ethanol. After that, I stirred the solution for around 10 minutes to allow the dislodgement of mites from the plant materials onto the solution. So after stirring, the next step was to filter the solution using sieves. Here we can see a stack of 3 different sized sieves. The one at the top has largest size pores, whereas the one at the bottom has the smallest size pores of 25 micrometer. I'd like to remind you all the size of the Phyllococcus fructifilus again. So it's 140 to 170 micrometer long and around 40 to 70 micrometer wide. So the sieve at the bottom had very small pores through which mite would not pass and would get collected at the bottom sieve. So after a number of washes, I rinsed the bottom sieve with again 7% ethanol onto the petri plate before counting them under the microscope. I repeated this procedure for 6 months. We started doing this on May and continued until October. I was just counting mites even before I came here. So the next thing that we did was collecting samples for RNA extraction and RT-qPCR to confirm the virus presence in our samples and to compare whether there's a relation between the mite counts and the virus presence. So for that, I collected samples, did RNA extraction, and did RT-qPCR to determine the virus presence. And here we go. This is the preliminary result that we have. I know the slide is very noisy, but this is the result of 6 months for all of the 12 cultivars. So the x-axis represents 12 different cultivars that we used in our study, and the y-axis represents the number of mites per gram tissue. Different color-coded bars represent different months. So after counting the mites, we could see the 9082 and 013, Tamu 730, Wichuriana and Knockout were the top 5 cultivars that had greater number of mites compared to other cultivars in our study. If we compare the mite numbers across different months, I would like all you to focus on the brown, blue, and this greenish bar, which represents the month of July, August, and September. So we had a higher number of mites during these months compared to the months before or after. And More or less, all of the cultivars had greater number during September, but for Camel 730, it was pretty much spread throughout the 5 months. Next, when I tried averaging the number of mites, for 6 months, it was the same 5 cultivars that had greater number of mites compared to others. And Bright Eyes and the pavement series had relatively smaller number of mites compared to other cultivars. In addition to that, we also noted disease severity in the mite population study field. So out of 12 different cultivars, only 4 had visible disease symptoms in the field, which you can see here. TAMU 730, Knockout 9082, and N13 were those 4 cultivars. TAMU 730 had almost zero severity in most of the plants. So there are 15 plants in total per cultivar. So in most of them, disease severity was 0, but for 1 or 2, it was really high, around 95 to 100%. When I averaged the percentage per cultivar, TAM 730 had the highest disease severity of around 13%, followed by 9082, and then N13, and then the knockout. So for RT-qPCR, we had collected both symptomatic as well as non-symptomatic tissue. And as expected, symptomatic and non-symptomatic tissues had different qPCR result. Even though the disease severity was around 95%, there was a difference. If I had collected a leaf that had no symptoms in it from the same shoot and another leaf that had symptoms, they had different results. So there was only one plant out of 180 that had the same qPCR results for both symptomatic as well as non-symptomatic tissue. So when I looked at the qPCR positive cultivars and tried to see if they align with the cultivars having the greater number of mites, it was pretty much similar. The only mismatch was Rosa wichuriana. There were no any plants in the field that had visible symptoms for this particular cultivar Rosa wichuriana. But for other cultivars, RT-qPCR positive cultivars align well with the cultivars having greater number of mites. So from this study, we could say that cultivars with low mite population and no rose-rosy disease symptoms included pavement series, Bright Eyes, Carolina, and Modern Plus. Other cultivars displayed varying degree of mite infestation and disease severity. So with this preliminary result, I would like to stress that I did not do any statistical correlation analysis, but from the data that we have got until now and through the graphs, we might say that tolerance to mites and the disease may be correlated because cultivars that supported lower mite densities also remained virus-free during the observation period. So I'll be doing the same thing on 2026 and 2027 to have a better idea of what's actually going on in the field. Just changing the gear, since I'm a PhD student, I like to point out other projects that we have been brainstorming to do. One of those is characterization of the rose mosaic virus. Since we all know it's a complex associated with single or mixed infection of viruses, it has become a threat to the propagating materials because it goes undetected in those materials. And we are planning to do non-targeted high-throughput sequencing, and hopefully we'll be able to identify known or novel viruses associated with rose mosaic disease. Of course, after characterizing, we'll have some genomic data. And at present, we all know that we need efficient diagnostic tool to support virus management. So down the line, we could use our data for virus indexing, as well as I was talking with Dr. Francisco about the possibility of EDNA as well. So lastly, I'd like to thank my lab members, everyone from Ranabhat Lab, North Lab, Clinton Lab, and everyone who are directly or indirectly involved with this project. Before I wrap up, I would like to mention that since I'm in my early career stage of my PhD, I would welcome all of your suggestions if you have any ideas or if you think I need to change something for my sampling period or my counts method or anything you have about mosaic characterization and everything, I'm all ears. Thank you.